Welcome to Legacy Academy. In this video, we are going to see about the Mendelian principle. Gregor Johann Mendel is the father of genetics. He conducted a series of experiments to demonstrate that the action of invisible factor, now called genes, in determining the traits of an organism. For that, he used a pea plant as model. He checked 7 different traits of pea plant, such as shape of the seed, color of the seed, color of the flower, form of the pod, color of the pod, position of the inflorescence, and finally the size of the plant. Now there arises a question why he selected a pea plant. There are some features needed to be checked before confirming a model of plant for genetics. These are as follows. The first one, it should have a short generation time, so he could get a result in next generation faster. The second feature is the easiness of pollination. It can be self-pollinated or cross-pollinated. The third one is the contrasting trait. As these traits are contrasting, the mixture of these traits in the next generation can be easily recognized. The last factor, that is large number of offspring. The large number of offspring increases the sample size. By the experiment with the pea plant, he made 3 laws, that are law of unit factor or the law of dominance, law of segregation or law of purity of gamete, and law of independent assortment. In law of dominance, he says that every character is controlled by a unit factor, and this unit factor exists in pairs. In case of dissimilar unit factor, the expressed one is dominant and the suppressed one is recessive. Here in the image, we can see that the capital letter P is represented for dominant character, that is purple, and the small letter p is for recessive character, that is white. In F1 generation, here it is a capital letter P and a small letter p, which is an heterozygous condition. In that, the flower shows a purple color which has the capital letter P, which suppresses the small letter p. Now we can understand what is law of segregation. In the law of segregation, factors governing a character remains together in an heterozygote without mixing or contaminating each other and get segregated during gamete formation. In case of law of independent assortment, the factor for one pair of character segregate or assort independent of the factor to the other character pair. Now we can check an example. For a character, T is equal to tall and small letter t equal to short. The other character, capital letter R equal to red color and small letter r equal to white color. So, an individual with homozygous tall character and a white color cross with an individual homozygous small character with red color. So these individuals will have the possibility of a capital T and small r gametes because both are homozygous condition. And this individual will have a small t and capital R condition gametes, and the same, they are homozygous. And these gametes can independently assort with each other to form a new individual. Now we can check some short formulas in the case of genetics. With the example, I will explain. In case of phenotypic group, to get the number of phenotypic group, we can take the 2 raised to n, where n is the number of traits taken. Example, in a monohybrid, there are one trait is selected. So, the number of phenotypic group is 2 raised to 1, that is 2. Two types of a phenotypic group. In a dihybrid, there are 2 types of traits, so 2 raised to 2, that is 4. Now we can check the same condition for genotypic group. In a genotypic group, it is 3 raised to n. So in a monohybrid, 3 raised to n means it is 3 raised to 1, that is 3. So 3 types of traits, that are 1 is to 2 is to 1, as we know. In a dihybrid cross, there are 2 types of trait. So 3 raised to n is equal to 3 raised to 2, that is 9. As we know, the ratios are 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. Okay, now we understand how we can get number of traits or number of groups by getting the number of traits in an individual. Now we can check number of gametes of an individual. To check the number of gametes, 2 raised to n is the formula, where n is the number of heterozygous locus. So some examples can be checked. In an individual having 2 T signs only, then what will be the number of gametes? Here, there is no heterozygous condition in this condition. So, 2 raised to 0, that is 1. In a condition T and small t, what is the number of gametes formed? Here, there is one heterozygous condition, so 2 raised to 1, that is 2. The other example, capital T, capital R and small r. Here, how much condition? Here only one heterozygous condition, that is capital R and small r. So again 2 raised to 1, that is 2. So we can check a larger one, that is in a individual having capital T and small t, capital R and small r, capital S and small s, and 2 capital W's. Here we can see 3 heterozygous conditions. So the number of gametes will be 2 raised to 3, that is 8. So we can get number of gametes by checking the number of heterozygous traits. Now we can check the probability in genetics. The probability has a main role in field of genetics. There are 2 types of rule, multiplication rule or AND rule, or the second one is addition rule or OR rule. In multiplication rule, we multiply wherever AND comes in the question. Here in this example, we can check that for an individual with heterozygous condition, capital A and small a, capital B and small b, to have a gamete of small a and small b, what will be the probability? To get a small a from this capital A and small a, the probability is half, because only one of 2 is a small a. The same is for small b. So half into or multiply by half gives 1 by 4. In the case of additional rule, Here, wherever the or comes, we add. Okay, in here the individual capital A and small b, capital A and small a, and capital B and small b. We want two type of gametes, that are small a and small b, and capital A and small b. So here we want two types, and here comes or. To have a capital A, here in this condition it is 1 by 2, and small b it is 1 by 2. When we multiply, we get 1 by 4. Or comes, so we need to write plus, addition sign. And in this condition, capital A, here only one capital A, so again 1 by 2, and small b, here only one small b, again 1 by 2, multiply, then we get 1 by 4. So we want to add this both 1 by 4, so we will get 2 by 4, and that is equal to half. So here the result will be half. So we can check few questions in this probability of genetics. So here a question in a cross, capital A, capital B, small b, capital C, small c. Condition individual, cross with small a, capital B, small b, capital C, small c, individual. And what is the probability of getting an individual of capital A, small small b, and small c, small c. So here we will multiply to get the probability, because here come ands, and we can multiply each of the traits separately. So we can write it separately first. Probability of getting capital A and small a multiplied by probability of getting small b, multiplied by probability of getting small c. Here we will get only capital A from in this individual, no, and not from this individual. So the probability will be 1, because only one individual have that, and one have only that trait. Multiplied by what is the probability of getting small one? Here only this individual have that trait, so here also we will write 1, and multiplied by the probability of getting small b in both, we will get from both, so the condition is 1 by 2. And again it is the probability of getting small b, so this again the value will be 1 by 2, multiplied by probability of getting small c. In both also it is a heterozygous condition, the same condition as 1 by b, it will be 1 by 2 multiplied by 1 by 2. So here the probability will be 1 into 1 by 4 multiplied by 1 by 4 gives 1 by 16. The probability of getting capital A, small b, small c from these individuals is 1 by 16. Now we can check a larger question which may be coming in competitive examination like CSIR NET. Mendel crossed tall pea plant with dwarf one. The F1 plants were all tall. When these F1 plants were self to produce F2 generation, he got a 3:1 tall to dwarf ratio in the offspring. What is the probability that out of these 3 plants of F2 generation picked up a random 2 would be dwarf and 1 would be tall? So for this condition, first we have to write the probability of getting a dwarf plant, that is 1 by 4. Here from this ratio we could find that only 1 dwarf out of 3. So total there will be 4 plants. So out of 4 plants there will be 1 dwarf. Then probability of getting a dwarf plant is 1 by 4. Then the probability of getting a tall plant is 3 by 4, because all the 3 were tall. So probability of getting a tall plant is 3 by 4. Now we can write the probability of getting two dwarf and a tall plant. So first condition, probability of getting first a dwarf, then a tall, then again a dwarf. Or it can be like probability of getting first a dwarf, again a dwarf and a tall. Or probability of getting first a tall, then dwarf, then dwarf. All these are the probability of getting two dwarf and one tall in from this ratio. So now we can find the probability of each condition. In first condition, it will be dwarf have 1 by 4 multiplied by tall will be 3 by 4 multiplied by 1 by 4. Or will be which sign? Or will be plus sign. Okay, then the second condition, dwarf will be 1 by 4 multiplied by 1 by 4 multiplied by 3 by 4. The OR condition will be plus sign. Then third one, tall condition 3 by 4 multiplied by 1 by 4 multiplied by 1 by 4. Okay, all this will be 3 by 64 plus 3 by 64 plus 3 by 64. So the probability of getting 2 dwarf out of 3 pulled from a F2 generation will be 9 by 64. And that's all for the Mendelian genetics. Hope you all enjoyed this video. So please subscribe, share and comment and follow us on Instagram.